Good afternoon. I'm the moderator of this session, Marina, Cunha. Can I ask the presentation first? So I was asked to do a small, short introduction to deep sea research for this session. This session is quite diverse, so I will have a general introduction to the deep sea. And during the debate, we will try to focus on the different topics that are going to be presented by the 6 speakers today. Okay? So I think that everybody in this, I will be quite quick with the slides, just for warning. Everybody knows why, in this room at least, why the oceans are relevant. There are some of the issues that why the oceans are relevant. And one of the most important things is that the oceans are deeply involved in the global biogeochemical cycles also. But why is the deep ocean also relevant, or even more relevant? Well, besides being the largest biome on Earth, the deep oceans provide crucial ecosystem services that are depicted here in this table. Oh, sorry, this is not on. Can we switch off the lights? I can't see the laser. Okay, that's it. So this is a summary of the main ecosystem services. Most of these are potential services. They are not actual services because some remain to be explored. But while we explore this ecosystem service by resource extraction or other human activity like dumping debris or pollution, we are also creating threats to the resources and to the functions of the ocean. Another source of stress is the climate change. And all these must be accommodated when we try to follow the route of the blue economy as we pretend. It's very difficult for us to assess impacts and risks in the deep ocean. And we know that there are several synergies and multiple stressors in the deep ocean. And in this picture, you can see some of the most important, the ones related with the climate change, like warming, acidification, and deoxygenation, but also related to human activities like fishing, oil and gas extraction, mining, or dumping litter or other types of pollution. How much of the deep ocean is protected? Well, according to these estimates, very, very little, only a little part of the deep ocean is protected. Only 1% of the ICs is under the protection of marine protected areas. And this is far from the IKE target 11 that predicts 10% of protected areas by 2020, next year. So we will not meet the target. Why should the deep ocean deserve more attention? Well, there's a huge knowledge deficit in the oceans. The open ocean is also the largest biome on Earth. And we know so very little about it. Less than 0.0001% is investigated in the deep water pelagic or even in the deep sea floor. So we have a very large knowledge deficit. We also have missing biodiversity baselines. Most of the ocean is not explored, and when we go away from the coast or deeper in the water column, especially in the water column, the deficit of knowledge on biodiversity is huge. What are the challenges? The challenges are various: societal, technological, scientific. There are several examples, but all becomes a challenge when we want to do deep sea research. Maybe one of the most important challenges is to show or make people aware of the vulnerability of the deep oceans. Because we call Earth the blue planet, the ocean planet. But in fact, when we put all the water together, it's only a small bubble on the top of the Earth. If we remove all water and put it in a sphere, it's very little. This small sphere of water contains 99% of the biosphere, more than 80% of the biomass, and from this, 70% is the deep ocean. So I think this image shows very clearly how vulnerable are the oceans, and especially the deep ocean that makes the most part of it. So we must say, and we must forward this message, that even the deep ocean is skin deep, in fact. It's not so deep as in our scale. So what are other challenges? We have to try to do this imbalance between threats and opportunities in favor of opportunities. So these are some of the most important threats and the opportunities that we have in the deep ocean. What we see now is that is a high lack of knowledge and poor governance. And this puts the balance towards the threats. But if we have scientific knowledge and strong institutions, good governance, we will have ocean sustainable opportunities. So I tried to find out what are the main deep ocean research questions, and I saw this summary by the Deep Ocean Steward Initiative, the DOSI. And this, in fact, is a summary, a very good summary of some of the most important issues that we have in the deep ocean. We are going to hear some of the results this afternoon about these issues also. What is the way forward? Well, the way forward, according to my opinion, it's that we have to have more networking, more multidisciplinarity, more spatial and temporal coverage. This map shows the inventory of sustained deep ocean observing. And as you can see, it seems okay in certain parts, but mind the scale. So there are vast areas of the deep ocean that are not observed in a sustained way. So we need more sustained observations, collaborations, and networking. With this idea, we have a great opportunity for the next decade. Is the UN decade of ocean sciences for sustainable development. And if you read these objectives, these priority areas, it's in a very good agreement with the questions that were identified by DOSI. And I think that in the framework of this conference, where we are trying to establish Atlantic-Mediterranean collaborations to see the interactions between the two basins, all this fits very well, and we have here in the next decade very good opportunities to establish such collaborations. So with further ado, I call the first speaker, Marta Álvarez. We have to run. Okay. Yeah. Can we have this your microphone?
afternoon, everybody. Okay, sorry. Good afternoon. I'm Marta Álvarez, working in A Coruña. Hello, Bella. Sorry. I'm in the Atlantic side, but I like to work also in the Mediterranean Sea. So I'm presenting CARIMED. CARIMED aims to be a data product to be publicly released, hopefully next year. It's been taking too long to accomplish all this data. CARIMED aims to be an internally consistent data product containing carbon and ancillary data for the different basins in the Mediterranean Sea. Here you can see the special coverage of the cruises we've been collecting in the different Mediterranean Sea subbasins, and you can see the big hole in Libyan waters. Our data expands since 1976 till 2018 with the last Medship cruise done in 2018. We are collecting ancillary, so hydrographic, inorganic nutrients, dissolved oxygen, but mostly we want to focus on CO2 measurements, variables like pH, alkalinity, and DIC, and also transient traces like CFCs and CF6. What we have been doing is collecting data from different cruises, digging in the literature, asking all the PIs, digging also in different databases to get different files that we merge and convert it to international units and to a common format that we are using Excel. After that, after collecting and formatting the data, we perform a first QC, so flagging especially the chemical variables with good or bad. And right now, what we are doing is what we call the secondary quality control. We want to assess if there is any systematic bias in the different chemicals variables, comparing the different cruises with a reference one that we presume is good enough to be compared with in layers where there is a minimum temporal variability. At the end, we want to release this product and get all the information uploaded to the different cruises in a table like this that contains, for each cruise and the different variables, all the information available and the correction factors that we applied or not. So if you want to know more about this effort, please come to my poster, and if you have data to share with us, please come to me as well. That's it. No questions.
Okay. So we will have all the questions at the end of the presentations. So I will introduce to you the next speaker, Menachem Goren, that will talk about the unique upper slope megafauna of the southeastern, western Mediterranean Sea.
Good afternoon. The Levant, the deep Levant Sea is not really tabula rasa, not complete tabula rasa, but our knowledge regarding the deep biota in this area is rather poor. Since the late 19th century, when the polar expedition visited this area and made some studying effort, very little has been done until the early 90s. Just to illustrate the lack of knowledge, I want to tell you that some serious scientists declare the Eastern Deep Sea a desert, a marine desert. And of course, this is not true. Just to show you what are the gaps in knowledge, when we started to study the fish of the deep sea at the early 90s, only 6 or 7 species were reported from the entire Levant Sea at that time. About 4 years ago, Professor Bella Galil and I published in Zootaxa a checklist of the deep sea, Levantine deep sea. We counted over 60 species there. However, 56 of them were along the Israeli coast. From other parts of the Levant Sea, we had records of only 5-6 species from each site. This doesn't mean that the Israeli coast is richer than other parts of the Levant Sea. It actually reflects the lack of knowledge and the very poor research effort invested in other parts of the Levant Sea. While in the deep sea is known to a certain extent, the continental slope along the eastern Mediterranean coast was really a tabula rasa. We didn't have any knowledge about the composition of the megafauna there. And we are very lucky that within the framework of EDEM project, you are invited to Google EDEM, after my talk I mean, we had the opportunities to study the continental slope, and I want to share with you some of our findings. First of all, for the methods, we had 8 cruises. We trolled over 4 depths. Each troll or each transect was about 12 kilometers, and altogether, each depth was dredged over of more than 1 square kilometer, which is quite a lot. What are the findings? First of all, you can see that we found 140 species. We have collected more than 100,000 individuals and 3,000 kilograms of animals. The richest taxa or group was the Osterichthys, the bonyfish, with 80 species, and then the mollusk, the crustacean with 20, and Chondrichthys with 10. We tried also to see the changes in the proportional biomass of the major group along the depth gradient. And as you can see here, where is the pointer? Sorry, opa. Okay, as you can see, the Chondrichthys, the first column, comprise about 1/3 of the biomass at shallow water, at 200 meters, and then their proportion decreases to 50% of the biomass, and then it goes down with the depth. Osterichthys, the bonyfish, comprise about 1/3 of the biomass at shallow water of 200 and 300 meters, and their proportion decreases to 55 to 60% when we go to deeper water. With mollusk, the story is quite different. Shallow water, we had about 25% of the biomass, but when we went down, we see that they are gradually disappeared. Crustacean comprise about 10%, 10% along the gradient depth. I'll try to make it short, just 1 minute. We tried also. If you need laser. Yeah. Both together? Okay. Okay. We tried also to see if there is any effect of depth on the body size of the animals, of the major groups, of the major species. And you see that in some cases we didn't find any difference. Raya clavata, this ray, shows that in shallow water, 1,200 grams. When you go to deeper water, it's about 1 kilogram, more or less, and 1 kilogram. On the other hand, you see that in cases of merlutius, in shallow water, it's 88 grams. Then we get to 266, 729, and then over 1 kilogram. So each species differs in its response to the depth. Of course, if you want to see more, you can see it downstairs on the poster, and to read some of our papers that will be published soon. But I can't finish without a very annoying slide to show you. This one. The bottom of the continental slope is covered with garbage, and you can see all kinds of things. Cans, drinking cans, concrete, sacks of concrete that were dropped, I don't know why, on the soft bottom and form a new habitat. Plastics of all kinds, not mentioning ghost nets and long lines. Thank you.
So for the next speaker will be, don't mind, Timor Katz, that will talk about the deep lab, the deep sea moored observatory in the Levantine Sea.
Okay, good afternoon, everyone. So I'm going to talk a bit about some results from Deep Lab, which is the first deep sea moored station in the eastern Levantine Basin, 50 kilometers away from land and 1,500 meters depth. Has many instruments, including sediment traps, current meters, CTDs, etc. So, what we see when we're looking at the mass fluxes is that we've got a very seasonal pattern, wherein the great majority of the flux comes in winter, and what we can also see is that there is a steep increase in the fluxes as you go beneath 180 meters. So what we are seeing is that actually most of the incoming flux is coming through intermediate nephloid layers that are coming somewhere between 180 meters and 800 meters. There's also a benthic nephloid layer, but this is not the main input. At the very bottom, near the seafloor, the currents are very slow, and there's no seasonal trend. Okay, it's the same winter and summer. Currents are much higher in the upper water column and do have a very seasonal pattern. Okay, so it's much faster in winter, but resuspension at the bottom is unlikely during most of the time. When we looked at the sedimentation rates. So the sedimentation rates at the study site, both as you calculate it from the sediment traps and whether you go and look at the sedimentation rate using radiocarbon dating, is about 100 grams per meter square per year, which is quite a lot considering the fact that the flux coming from above is really small, and about 0.5 to 0.6 grams of organic carbon are getting buried per year in that area, and this is actually most of this carbon is coming with this lateral flux rather than from above, and about a third is getting buried, which is a lot considering, and this also has to do with the fluxes from the sides. So when you're looking at the sediments, and if you're looking at stations going away from shore, you can see that geochemical signatures of proximity to the shelf do decline as you go away, but the effects of this lateral transport would go to at least 100 kilometers away from shore. So we can see these effects going further and further, and as you go further, of course, sedimentation rates greatly decrease, and Again, we're talking about effects that go way out. That's it. So, conclusions. The new observations provide initial insights to the East Levantine Basin in all kinds of aspects. You'll hear more about it. At least 3/5 of the sediment flux that reaches the seafloor comes from the side, from particle export from the shelf and from the land. This lateral transport occurs mostly in winter and is probably due to either, or more likely both, storms and floods coming in. That's it. And measured bottom currents were constantly weak, so there's not a lot of resuspension, although there is a small benthic nephloid layer sometimes observed. We calculate sediment accumulation rates in deep level of approximately 105 grams per meter square and 0.55 grams of carbon per meter square per year. We've said all that. Lateral transport effects, or off the continental margin effects, to at least 100 kilometers. That's it, and thanks to the supporting groups, and thanks for listening. You can hear more about it later.
So the next speaker will be Suzan Kholeif. She will be talking about the dinoflagellate cysts in the Nile deep sea fan.
Yes. Good afternoon. My name is Suzan Kholeif. I am a professor of marine geology and former president of National Institute of Oceanography and Fisheries. What's this? Where?
You just move.
Right one?
Yeah, the other one. Okay.
Yes. Sorry. Excuse me. My study or my papers about an overview of dinoflagellate cysts population in the Nile deep sea fan. What is Nile deep sea fan? Is the biggest fan, is the biggest structures in the Mediterranean Sea, is originated or built during the Messinian crisis 30 million years before present. Characterized by numerous fluid seep structure outcropping in the water depths, including big mark, including a mud volcano, and ancient seep system or biodiversity. And these samples collected during a meteor cruises 2006, and the aim of the study. Sorry. Where is the aim of the study? Sorry, I missed. The aim of the study is to study the systems, the flora or fauna can be live in this strange ecosystem, and this ecosystem located at depths start from 1,000 until 3,000 depths in the Mediterranean Sea. I don't know this, not my presentation, it's not last one, this aim. So we didn't expect that we find any flora or fauna in this system, because the system mainly covered by H2S. And you couldn't find, we don't expect to find any flora or fauna inside. However, we have amazing result. We find about 45 species of dinoflagellate cyst. The first group is indicated the heterotrophic environment, and like that. Sorry, I don't know how can like that. Heterotrophic, and another one is indicated of the freshwater seepage in this area, and this freshwater from the Messinian indicated that the Nile River at this area, at these ages, can flow and the old Nile River branch located at this area. And we find also algae, freshwater algae, and and also oligotrophic spinophorite ramosis, and associated with operculum. We expect that this oligotrophic transport this area because this area expected to all heterotrophic because the gases H2S and CO2 and the bacterial mat cover all this thing. I'm so sorry, but this is not all my presentation, this old one. I put some figure of the sediment to show you what the true photo to show this ecosystem, because this ecosystem is amazing and the unique in the Mediterranean, and they need more effort to study. And only this cruise by Meteor 2006 start to study this ecosystem. So I am so grateful to Max Planck Institute team and Geoscience Azor of France for financing me to join this cruise and pay for collect sample. Thank you so much.
So our next speaker will be Toste Tanhua. And Toste will be talking about the time series data of stable isotopes and transient traces.
So thank you very much. So I'm going to talk about the deep ocean ventilation of the Mediterranean. As you know, the Mediterranean is pretty deep, 4,000 to 5,000 meters, and it's characterized being very well ventilated. I'm going to show some data in time series of data that going to describe a little bit of how this ventilation has evolved over time. So this map here shows all the positions where I have carbon-13 data. So that is the delta carbon-13. So some of the black dots here is from Catherine, forgot the name, sorry about that, but somebody in the 1990s was thoughtful enough to take a lot of measurements of stable isotopes and publish those data, so it's available now. And we have data from 2011 and 2018 that we can compare to. So first of all, we will look at transient traces, and this here is the atmospheric history of CFC12 and SF6, and you can see here from 1930s, 40s, how did they increase in the atmosphere over time. These are anthropogenic compounds, and they are equilibrating with the seawater, and then the seawater moves from the surface to the deep ocean. So if you find high concentrations of CFCs in the interior ocean, it means that it's well ventilated, and if it's low concentration, it's less well ventilated. And what you can see is a very interesting profile. This is from our cruise in 2018, and this is the SF6 data. So it shows you that in the Western Mediterranean, we have a big section here of newly ventilated deep water. And you have an area at 1,000 meters about that is less well ventilated. And this is a unique feature. You don't see that kind of profiles of transient traces anywhere else in the world. And if you start looking at this section and the sections that have been taken previously, you're going to see how the ventilation of deep Western Mediterranean has increased the last decade, that we know well from other work by Catherine, for instance. So, and when the water leaves the surface, it takes with it perturbations, such as uptake of anthropogenic carbon. So the Mediterranean stores large amount of anthropogenic carbon in comparison to its surface. And the reason for it doing that is the higher well factor. The deep water of the Mediterranean is roughly 13 degrees. It's a very high salinity and alkalinity. These factors brings it a very high potential to take up extra carbon. And by the very fast ventilation, that is actually getting from the surface down to the deep ocean and getting stored. So the Western and the whole Mediterranean is a very important part for storing anthropogenic carbon. And when we're burning fossil fuel, we change in the stable carbon isotope relation of the atmosphere. because there is a fractionation of carbon isotopes by photosynthesis, and oil is coming from plants. So you're decreasing that ratio, the carbon-13, right? And that is then transformed into the ocean. So you can see some graphs here how an increasing DIC leads to a decreasing of carbon-13. Now we're lucky enough to have all this data, and I invite you to come to the poster. We'll discuss the details of this. But you know, this graph here, for instance, is the delta C13 of the Adriatic Sea, the Adriatic Pit. The black line is the 1988 data. The red line is the 2011 data. And there's a big difference for the whole water column of carbon-13, which can translate to an uptake of anthropogenic carbon for the whole water column. And this is kind of unique. You know, you don't really see that kind of big impact for the whole water column anywhere else on the planet, maybe in a Labrador Sea, but this is a very big uptake. So what is interesting here is that when we look at the Western Mediterranean, we see a very different thing here. In the Western Mediterranean, for instance, here, the blue line is the 2018 data, and the red one is the 2011 data. And you see an increase in carbon-13, which actually translates to a negative anthropogenic carbon uptake. And that is inconsistent with the ventilation information that we have. And we are starting to try to understand what this actually means. Is the Mediterranean taking up less anthropogenic carbon than we thought it did, based on the CFC ventilation data, or is it some other interpretation thing that we have to do with different water masses and changes in water masses? So this is very much a work in progress, and I invite you to come to the poster and discuss this with us. Thank you.
Now for the last speaker of our session, Katia Bartilotti will talk about the zooey of the stripped soldier shrimp.
Good afternoon. I only have 2 minutes, so I'll try to be really fast, and I have to invite you all to visit my poster. Otherwise, you don't understand most of the things that we have here, since they are related with figures that I have in the poster and not in here. But the most important message of this poster is that we are trying to fight the unknown, and in this case, we are trying to do it studying the first 10 zoal stages of the stripped soldier shrimp. This is a species with an economic importance in the fisheries in the Mediterranean Sea. And the objectives of this work are to understand reproductive biology and the offspring development of deep sea shrimp, to redescribe the first 7 zoal stages of this species, since it was already described 10 years ago, and to describe the 8th to the 10th zoal stages of the species. After 29 days, we were able to get 10 zoal stages, and in the first 7, comparing it with the previous description, we saw only small differences. Although, for the first time, we described the 2nd and the 3rd priopod, since the previous author was not able to do it, probably because he had a few number of larvae. In the 8th zoal stage, we had an antennal flagellum as long as the antennal scale, and the 3rd priopod was already enlarged in size. In the 9th zoal stage, we had an antennal flagellum longer than the scaphocerate, or the antennal scale, and the priopods 1 to 3 were functional, but the 4th was present as a bud. These are the things that I have to explain you with a figure. In the 10th zoal stage, we already have an antennal flagellum 2 times longer than the scaphocerate, and the priopods 1 to 4 are functional, but the pleopods are still absent. This means that probably this larva needs at least 3 more stages to complete the development until the decapodid stage. Although, and even knowing that this genus has more than 90 extant species described to date, this is the longest larval series for the genus obtained under laboratory conditions. Thank you very much.
So thank you all speakers for keeping your time. We have exactly 20 minutes to the end of the session. That was the predicted time for discussion. So I invite everybody to come forward and ask nice things to our speakers. Any questions?
Hi, to Toste. I would like to hear your thoughts as the Mediterranean is definitely warming. What would be the impact of warming on deep ventilation in the different basins?
So it's a good question. It's actually getting saltier as well as getting warmer. So the density is not changing that much. It's more or less constant or maybe slightly increasing in ventilation. So the important thing is the stratification. What is the change in density of the water column? And we see in the shift from strong shift in ventilation in Adriatic to the Aegean and then back to the Adriatic, and then we have the Eastern Mediterranean transient signal coming to the Western Mediterranean, starting in the Western Mediterranean transient, I think it's called, right? So I mean, I think there are a very complex situation of processes, and I don't see a decrease or a change in ventilation coming up. But when you see the poster, there has not been any ventilation at all since the Eastern Mediterranean transient in the Levantine basin. We see changes in S&T, but the CFC concentration stands constant in the whole Levantine. So really no change, no new deep water formation in the Eastern Mediterranean. Well, I don't know. I mean, it's really good to have carbon coming into the deep ocean. It takes it away from the atmosphere. It decreases pH, but I don't think there's a big deal in the deep Levantine. So I think there are things that we need to understand. There is changes in the Ionian Sea, so we see the outflow from the Adriatic coming to the Ionian, but east of the Ionian we don't see anything.
Thank you. The same authors, following the Professor Galil, decreasing the pH, you said? Decreasing. Yeah. Yes. Means more acidification we expect or what? Yeah.
Okay.
Thank you.
So carbon dioxide, when it dissolves in seawater, it's an acid and it decreases the pH. So if you have an increased uptake of anthropogenic carbon, you do decrease the pH of the seawater. That's ocean acidification. But since the Mediterranean is highly buffered, has high alkalinity and so on, the other factors like the saturation of aragonite is still pretty high. So there is a big resilience in the system towards acidification of the Mediterranean, I think.
Okay, thank you very much.
Also to you. So just, has there been any, okay. Has there been any changes that you know of cold water cascading to the deep sea, as has been shown in the Gulf of Lyon, or other things that may affect ventilation of deep sea?
I'm not sure. I know there are in the Adriatic, there were some very cold winters where we have very strong deep water formation, but when it moves, so that was northern Adriatic, but the data I have seen so far in the southern Adriatic, you don't really see that really cold temperatures going through. So it seems to be moderated by the intermediate layers in the Adriatic. So once it passes the Strait of Otranto, you don't see that very cold signal. But I mean, it's event-driven, right? So you do have influence of cold weather. And if you have a salty water, then you can create this high density.
Maybe if you get warming of the surface water, you can get easier cascading.
No, you need to get the cooling of the surface water. So you need that, the Boras effect in the northern Adriatic and so on. So what we're looking at is the transient traces. So this is a kind of integrated signal over many years. So it doesn't really tell you much about the processes behind it. So there are physical oceanographers here that actually can explain that much better than I can. What we're looking at is the integrated effect on the basin, the central basins. Yeah.
Any more questions? No? Maybe I have a question for the biologists. So the three of you. Could you explain or show what is the importance of having a good spatial and time series coverage of the environmental conditions for your work?
For my work, the most important thing is to have a baseline. So we have to first identify what we have, I think, before mapping or whatever. We can do both things at the same time, but these larvae appear in the zooplankton samples, and I was looking at zooplankton samples of the colleague of the Balearic Islands that were collecting, and they were the ones that were rearing the larvae. And we were not able to find any of these Plesionic edwardsi species. And maybe this is one of the most abundant there. So in fact, we don't know anything because the ones that were more abundant are not identified yet. So we have to first understand what identify and then, or maybe both things at the same time.
Go further in the biology and
Yeah.
then try to find some environmental explanation for for the distribution.
Yes, we saw this for other species. We were presenting a paper recently with a more deep species like that no one cares, but it's a very interesting one in the biology perspective, and in the even if we look at the environment where this species lives, all the evolutionary conditions and everything might be understood with the kind of studies that I'm doing. So,
from your presentation, take some words. Deep sea ecosystem still need more effort, and from all of organization, from all of scientists. For example, this studies, the first one to study in the Mediterranean for biological, at the base of biological set. We need more crews, more sampling, more study to for deep sea sediment, deep sea ecosystem, because it's neglected by many scientists because the money, because it's very expensive to collect sample, and you need crews, you need big expedition to collect, you need ROV. It's so expensive to collect this sample, but we need effort from all international organizations to help us for more study, for more of the deep sea ecosystem.
I think that she says everything.
So, may I presume that the knowledge on biodiversity is so scarce that we cannot go a further step on and try to relate with the environmental time series and all the effort that is being done with the observatories, this effort for having long time series and a better spatial cover. We need first to go on the biodiversity knowledge. Is that your feeling?
Yes, absolutely. Well, we are very lucky that in our area, the Institute of Oceanographic carried a lot of study recently on environmental conditions. So we gather, as you can hear later, a lot of information, but regarding biodiversity, it's expensive, it's need expertise, and the Levant Sea suffer of both.
Can I have the perspective of the people that are looking, maybe from your mooring observatory? What can you do for the biologists?
I'm sorry, I'm a geologist, but well, just this is really a strange thing that happened to us, which has not been recorded. But our biggest problem in 1.5 years of running this thing is that a Barcudina species, which was hardly mentioned anywhere, swarmed in big numbers into the sediment traps, reaching up to 40 individuals, about 15 centimeters long, getting into one bottle in each time. So we just, near Stern and et al. Just published a paper about this pelagic species that very little was known about, just because they ruined our experiments. So just about the biology that you find out about without intending to.
So, but for instance, do you have sediment traps on your?
Yeah. There are sediment traps. There are people working on it. There's.
Do you find larvae, for instance, for
well, not so much. But there's a big work about foraminiferas that has just came out, and but again, I don't want to get too deeply into things I know little about, so I'll leave it to the people who did it. Okay.
Maybe a question for Marta.
Yes.
What is the importance of having these consistent data products?
Well, thank you for asking that, because I didn't mention in the introduction. There is a lack of inorganic carbon measurements in the Mediterranean Sea. As Toste has just said, this basin is a laboratory basin for ocean acidification studies, because it really is prone to uptake anthropogenic carbon, but not only uptaking that and bringing it to the surface, but introducing it into the deep ocean because of the fast overturning circulation. So if we want to know how much of this carbon goes into the deep ocean and how much this basin is acidifying, we need data. And data publicly available and easily findable is really difficult. We've been struggling to get all these 40 cruises formatted and merged and in a easy format. And yeah, that's the importance of this database. I mean, publicly available data is very important for the oceanographic community.
From this small debate, I think that we can assume that there's not much communication between different research fields, different scientific fields. Maybe it's time to get together. Another thing that, I don't know if there's some questions.
I have another question to Toste regarding the C13 anomaly. Can you share us with some thoughts that you have? Why do you see this anomaly? Is it maybe affected by methane fluxes or by heterotrophic activity in the different basins?
Thank you. I'm not quite sure, Barak, why we see that strange things in the Western Mediterranean and in the Alboran Sea. And it's something that we are thinking about. I think it has to do with the intense deep water formation in the Western Mediterranean, how you moved up relatively old water up in the water column, and that is now moving towards the Alboran Sea. Why that is not quite reflected in the transient traces, I'm not quite sure yet. So actually I'm looking forward to Marta to have the CARIMED DIC consistent database to see what are the changes in DIC and oxygen. So these are the things that we're going to start looking into. Thank you. Then I also would like to say what I reported on and what Marta has been doing is kind of repeat hydrography program. So that is a deep sea ocean observing system like DOSI and DOOS. And that would be really interesting for the biology community to see what can we do on our deep sections, taking CDD, and to actually support consistent, sustained ocean observing of biology. I think this is important. You talk about getting a baseline, which is correct, but we need to think forward. How can we actually see what did it change? And how does it change things? But if you don't have a baseline, it's obviously hard to see changes, right? So I think, yeah, I don't know how.
Okay, so just one thing that I wanted to say is, as we said, the deep lev is actually the first deep sea, and then as we showed, it doesn't behave exactly like a deep sea station, but it's the first deep sea station in the Levantine Basin, and it is a huge struggle to keep it going over time. And we are struggling to keep it in place, and we're scraping. So the difficulty is, as you say, of course, the interdisciplinary work and to get the data out there, but even to just maintain these things over time, especially when things are changing so quickly, is a big issue that needs to be discussed, how to keep these things running.
So this is a transversal issue, funding and also sustained funding to keep these observatories, because I think that's the way forward, is to have these sustained observatories. We need to fill the gaps, and this is probably the only way. For the biology, it's important to try to determine what kind of variables can we measure in this sustained way, to try to have also time series of this biological data. I think, okay.
Thank you very much. It's a very interesting session about deep sea. I have a reflection about the microbial loop, as it's very essential to explain all the process of heating, of uptake of carbonic gas, and so, and that really we still have more efforts to deploy for explaining what it is, the reality of metabolic and changing in deep sea, and we have to put on more and more cruises like geotrace, for example. It's very important, and it's a recommendation to have a reflection, summarize reflection and interpretation in real time for all what it happened. Then, for example, for carbonic gas, for nitrates, and so we have to consider the microbial loop in real time. Thank you very much.
Just we are approaching the end, and I don't want to finish this session without asking the speakers, what is the added value of collaboration with scientists working on the Atlantic? Anyone?
Yeah. For my point of view, as oceanographer or scientist, we need to focus more on multidisciplinary oceanography study or science, and more communication and transboundary communication between countries. So imagine that the one paper was all to the 4 paper or 6 paper to put on the one concentrated research paper. It will be amazing result. Yeah, we need more new frontier of oceanography science.
Apart from the obvious connection in the outflow and inflow from the Mediterranean, I think to share best practices in the community on how to do observations and do science, and it comes to data management and information, I think there we can win a lot from the Mediterranean and also the Atlantic community, say, to share best practices and agree on best practices so we can compare observations in the Atlantic with the ones in the Mediterranean.
When you are saying best practices, are you talking about standardization and standardized procedures, measurements?
Yes, that's what I'm talking about. It's not a standard in a way that is prescribed how you do it, but the community agreed on best practices. There is a best practices system building up at IOC, UDE now, and I recommend you to look for those best practices on how to conduct a different part of the ocean observing and ocean science systems.
I'm sorry. Very short. questions to you. Yes, your presentations. You said only 1% in the high sea is under the protections.
Yeah.
Where is it? Which region?
It's scattered. Sorry, I didn't show the map of the areas that are protected, but considering the large surface and volume of water in the ices, even if there are large MPAs, this is a very small part of it. There are
That was from the Mediterranean Sea or that from the whole world?
from the whole world.
Maybe northwestern Atlantic.
Yes, in the northwest Atlantic, there are, for instance, in the Clarion Clipperton zone, there are 9 protection areas. There are several areas scattered all around the world, but it's not reaching much more than 1%.
Okay.
The Azores, yes.
I don't want to discuss more this issue, but according to me, CIESM already published one book, as you know, Deep Sea, Known, Unknown, by Tanhua, etc. I think it's one of the results can be from this work, discussion, we need one capacity building workshop or entire Mediterranean Sea, West, Central and East, I believe. Because we need the young scientists for deep sea. Thank you.
So we have no time, but I must remind you that we will follow with the second deep sea session, and my colleague Luis Menezes will moderate the next session, so we'll have other opportunities to continue this interesting debate. Thank you to all.